Okay, so let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us, the 64th seminar, and I call it a WashU day. After this seminar today, I'll be again out of town one last time this year, and this trip, the last trip, will make my out of town days more than 160 days with the 68 presentation this year. And I am looking forward to Christmas holiday break without any trip this year. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Michael Brent, my colleague at WashU. In fact, I met him 2011, 11 years ago or more, when I actually here for the interview, and he was one of my meeting person during that interview days. And he's a professor at computer science, genetics, and biomedical engineering. He keeps saying to me his synthetic biology experience is minimal, but to me, he's one of the pioneers who combine quantitative analysis of biology or modeling of biological systems with the experiments to understand the living system's complex metabolic and regulatory networks. Such a combination is actually the hallmark of synthetic biology. That means he's one of the contributor to this field very, very early in this field. So, Michael, this is my great honor again to have you today. And thank you so much for your time today among your busy schedule. And the virtual podium is all yours. And thank you.
Okay, thank you for that very generous introduction. I'm going to share my screen here. You can see my slides?
Yes, wonderful.
Okay, so Taesuk asked me to share a few thoughts about my career, and I will kind of start with the conclusions. My reflections on my career so far are that I value learning new things, especially mathematics and its applications, above being an established expert. I have a restless intellect. I've taken risks and opportunities associated with moving into new fields on multiple occasions, including a brief foray into synthetic biology. These choices are part of the reason that I have never become a grand old man in any field. I've always been a newcomer and an outsider. Yet I don't regret these choices, as they've enabled me to travel far and wide and to experience intellectual stimulation in diverse fields. I began my career as an undergraduate at MIT, and when I applied there, my intention was to become a gravitational physicist, but I ended up with a degree in mathematics. I had always been interested in learning foreign languages, and I heard about machine learning from a friend of mine, and I joined a research group as an undergraduate in computational linguistics, took courses in computer science and linguistics, and applied to the computer science PhD program. I had the great good luck that I had a champion in my research advisor, and I was admitted to the program, even though my application was not as impressive as some other people. So as a PhD student, I focused on machine learning of human languages. And while I was there, I developed a side interest in molecular biology and took some biology courses, but continued to focus my research on computational linguistics. At the end of my dissertation period, I began to think about how humans learn their native languages. I applied to exactly one faculty position in cognitive science at Johns Hopkins University, and I was very lucky that they offered me that position. I think I was just in the right place at the right time with the right advisor, and I was offered that position. I stayed at Johns Hopkins through assistant and associate professor of cognitive science, and I focused on computational models of how children learn language. I found great mentors who helped me get into that field, which was tremendously lucky. And I published in journals such as Cognition, and even have one paper in the Journal of Experimental Psychology. I have published several papers at that time that are still frequently cited, even 25 years after their publication. So I'm not a very proud person, but that does give me some satisfaction that people are still thinking about the things that I wrote about then. I married a biochemist, which was great good luck. And we did a national job search for 2 faculty positions in the same city. Her position was in Manhattan, and together we landed at at Washington University in St. Louis, which was at the time when it was one of the major centers contributing to the initial sequencing and analysis of the human genome. I was still interested in biology and decided this was an opportunity to tack back that way. I interviewed in Washington University computer science department based on my language research, and I was very lucky that they were broad-minded and offered me the position, even though my work was quite peripheral to the core of computer science. Rather than staying in developmental psycholinguistics, where I was established, I took the riskier option of trying to transition into genome sequence analysis. At an abstract level, language learning and sequence analysis have some common mathematical underpinnings, probabilistic models like hidden Markov models. So there was that little connection between the two. I wasn't coming into it completely with no relevant background, but it was quite abstract. And I was fortunate, again, to be able to recruit a postdoc who was in the Genome Sequencing Center here at WashU and wanted to learn more about computational methods, and a new graduate student in the biomedical engineering department. Without the help of these 2 people, I would probably not have been able to get into this field. And I wrote an NIH proposal and got a grant to do genome sequence analysis at a time when the funding rates for NIH grants were almost 25%. So again, I was fortunate, and I was in the right place at the right time with the right idea. And since I came to Washington University, I was promoted to full professor and then given an endowed chair. And I was lucky to have a department chair who was supportive of me and my various endeavors, even though I was not central to the discipline of computer science. Once I got into the human genome sequence analysis and the mouse genome sequence analysis, I stayed in that area for 7 or 8 years. I was able to develop a simple wet lab capability that was funded by the National Human Genome Research Institute. And I was invited to move my lab from the main campus in the computer science department over to the Center for Genome Sciences on the Washington University School of Medicine campus. From roughly 2008 to 2019, I focused on regulatory systems biology, especially using yeast as a model system, and a little bit of synthetic biology. And in 2019, I began another turn. I was invited to join an established research consortium studying the genetics of exceptional human longevity. And I was asked to do this because I had some expertise in RNA-seq analysis, and they wanted somebody who could provide a systems perspective. And so I was again in the right place at the right time. And I'm now moving into human genetics, and particularly in the area of multi-omics integration. Will I succeed and establish myself here? I don't know. It's still an open question. So to summarize everything that I've said, my career path has been shaped by my passions. My successes, such as they are, required a lot of good luck. Good decisions, skills, and abilities are necessary for career success, but by no means sufficient. And I think young people sometimes attach too much importance to staying the course and succeeding rather than adapting to the challenges and opportunities the world presents. I am often reminded of a study that I heard of, which said that 3 years after a tenure decision, faculty members who were denied tenure we're just as happy as those who are granted tenure. And my conclusion from that is that we are more adaptable than we realize and expect. Thank you.
Oh my goodness, amazing. So, I completely agree with you. And you keep talking about log. I believe in log, but log is only coming to us when we constantly seeking for it. Of course, I mean, you took the whatever opportunity, even though that was risky. So that's why you mentioned you have luck, but I guess that's not necessarily just luck, because you constantly seeking for new opportunity and then you took it. So yeah, that is amazing. I even didn't know. I mean, your original one was I remember, your paper when I interviewed, because I searched for all the papers, whoever I'm meeting, and then I remember now you did some linguistic work earlier, but I didn't connect how that kind of lead to this genome sequencing and the synthetic biology field. So that's amazing. So yeah, thank you so much. Yeah. Okay. Now the main speaker of today with a longer introduction, Dr. Pinglay is a postdoc at NYU School of Medicine, working toward engineering biological systems in order to understand their function. In particular, he constructs embryonic stem cells bearing large pieces of synthetic DNA to investigate how genes are tuned on and off through development. I kind of find this is a very interesting coincidence because Michael basically participate in early sequencing effort, at WashU, and then that basically allow us to move to the next stage. That's basically synthesis of DNA. And then now Dr. Pinglay basically using synthetic DNA to understand biological system better. So this is kind of generation to generation knowledge transfer and then making innovation happen. He's from Bangalore, India, and completed undergraduate degree in molecular biology and philosophy at Johns Hopkins University. That's another interesting part because there are some common university now I see between Michael and him. And when he is not dealing with DNA, he enjoys soccer. I believe he meant football, heavy metal, guitar, food, and sharing the joys of scientific worldview with others. I believe with the FIFA World Cup becomes more exciting with the knockout stage. I guess we need to compete with people watching the game over the seminar. But I believe, many people will watch this one today and over the Zoom, the YouTube video. So Dr. Pinglay, thank you so much for your time today. And please take it away. This is all yours now. Thank you.
Great. Thanks, Taesuk, for that very kind introduction. I don't think I'll ever be able to compete with the World Cup, but I'm happy to draw a couple of eyeballs away from it. And thankfully, there's no game today. Okay. Are you able to share the screen? Oh, sorry, see the screen?
Yep.
Fantastic. Awesome. Cool. So what I'd like to tell you about today is really the bulk of my PhD work, where we applied our ability to build really large synthetic DNA molecules to understand how genes are turned on and off in mammalian cells. Now, given that this is a synthetic biology seminar series, it wouldn't be complete without someone saying, what I cannot create, I do not understand. Yeah, and that's really kind of the philosophy that underlies this entire approach. Right? We want to try to build complicated gene regulatory principles from the ground up in order to understand how they function. And what I'll tell you about today, majority of what I'll tell you about today is looking at one specific region in the genome, which is called the Hox cluster. But as a means of introduction, what I'd like to tell you about, just to kind of set the stage, is protein coding genes only make up something about 2% of the human genome, as became clear after the Human Genome Project. Now, you might wonder what is the rest 98% doing? And over the last 20, 30, 40 years, it's become clear that this non-coding DNA really serves as a blueprint for when to turn on and off this 2%. And because the regulation of gene expression is of critical importance for development to respond to disease states and things like that, it's often relies on relies on multiple regulatory elements. And I just want to quickly tell you what these kinds of regulatory elements might be. So we've gone from this kind of single gene view to a very complicated view where there's many, many, many different kinds of elements that are coming together to give us a particular gene expression pattern. So for instance, let's just say there's this gene here that's expressed in our favorite cell type, and there's this neighboring gene here that's not expressed. Now, that could be for a few reasons. One, this neighboring gene may be decorated with something like inactive chromatin. Secondly, there are elements called enhancers in mammalian genomes that can be located really far away from their target gene. We're talking about hundreds of kilobases away. And these serve as binding sites for transcription factors, which then are thought to kind of loop into the promoter of that gene and control transcription. Now, you might be wondering why, for example, there are certain enhancers that are located closer to this neighboring gene, but seem to activate our favorite gene here in green. And the reason for that, and some hints are coming from looking at the three-dimensional organization of chromatin in the nucleus. And it turns out that in lots of complex genomes, the three-dimensional organization around genes is structured such that these enhancers are preferentially interacting with the promoter of its target gene, but somewhat occluded from neighboring genes in these domain structures called topologically associated domains, or TADs. But I think what we really want to come to an understanding of is how these multiple layers of regulation are coming together in order to give us this gene expression pattern. And if there's one thing that I'd like to tell you today is that our genetic engineering technologies to perturb all of these multi-way connections need to keep pace with this kind of genomic understanding of gene regulation. So we can look at the whole genome, but to make it a little bit simpler to start off, what we thought we could do was focus on certain model loci that have served as models for the study of gene regulation. And one of those in mammalian cells has been these sets of genes called the Hox genes. So Hox genes are a set of deeply conserved transcription factors that pattern the head-to-tail axis of the developing embryo. Now, in mammals, they're organized in 4 clusters, A, B, C, and D, and each of these clusters contains a subset of 13 genes. Now, the remarkable thing about the organization of these genes is that genes found earlier in the cluster are expressed closer to the head, whereas genes found at the end of the cluster are expressed closer to the tail. Right, so you can see here, so HOXA1 here would be expressed closer to the head in the developing embryo, and HOXA13 would be at the tail. Now, this kind of remarkably showcases the fundamental link between genome structure and controlling gene expression. And therefore, since they were initially discovered in mammals about 40 years ago, they've become a model for the study of gene regulation. And every time we learn about a new layer of gene regulation, we also find it at the Hox cluster. And for the purposes of this talk, we're going to focus on the HOXA cluster that plays a very important role in specifying the identity across the nervous system. And to study it in great detail, we have the benefit of an in vitro differentiation system that makes use of mouse embryonic stem cells and converts them to motor neurons. Okay. So in the next series of slides, what I'm going to show you here is on the top where we are in this differentiation system. And on the bottom, I'm going to show you a schematic of the molecular events that are happening at the Hoxsey cluster. Right, so in mouse embryonic stem cells, or ES cells, none of the Hox genes are expressed. And the entire cluster is decorated with repressive K27 trimethyl marks. Now you can take these ES cells and treat them with differentiating signals, just as they would experience in the developing embryo. And one of these is this signal called retinoic acid. And in response to retinoic acid, a dramatic reorganization occurs at the Hox cluster. You get the splitting of the cluster into an active and inactive domain of gene expression. So you get, in response to retinoic acid, HOXA1 through HOXA5 expressed, and the rest of the genes not expressed. You also get a splitting at the level of chromatin, right? So you get the heterochromatin domain restricted to just those genes that are not expressed, and you get euchromatin from A1 through A5 that are expressed. In addition, what's correlated with this is the binding of the retinoic acid receptor, which is a transcription factor that's downstream of retinoic acid, and it binds all the way up to about HOXA5, which correlates nicely with this boundary between active and inactive genes. Okay. Now, in addition to things that are happening within the cluster, we have a whole bunch of things, whole bunch of regulatory elements or enhancers that get activated upstream of the cluster that loop in and control Hox gene expression. Now, you might wonder why these enhancers are restricted to just acting on those genes in the active domain, but don't activate any of the genes in the posterior domain. And if you guessed three-dimensional organization, you're right. And what happens is in 3D, the enhancers are given preferential access to those genes in the active domain, whereas they're somewhat occluded from those genes in the inactive domain. And if you go in and make genetic changes that manipulate the chromatin organization, you actually now get misexpression of more posterior genes, right? So I know none of you guys are developmental biologists, but the point here is to kind of just give you a sense of how complicated this is, right? And if we want to understand how these different layers of regulation are coming together, right? So you have internal transcription factor binding, we have distal enhancers, we have dynamic chromatin domains, and we have three-dimensional organization. If we want to understand how these different layers are coming together, that's a quite complex problem, and that's what we really want to do. So just taking a step back and thinking about why understanding gene regulation is hard, I think it's for two reasons. One, I think we lack the tools to manipulate multiple elements on the same piece of DNA at the same time. What do I mean by that? A lot of you might be thinking, hey, we live in a CRISPR age. Why is it difficult to manipulate DNA in cells anymore? But here's why. Even CRISPR only goes so far, right? So this is just a schematic of the Hoxsey cluster where I'm showing you enhancers here, showing you where the transcription factor binding sites are within the cluster and the position of the chromatin domain. Now, let's say I wanted to ask a question of what happens if I get rid of all instances of this transcription factor binding site within the cluster. Now, you could potentially ask this question by CRISPR, but the way you'd go about it is you'd go in, make one CRISPR edit, get your cell line, go in, make another CRISPR edit, make your cell line, and so on and so forth. And by the time you got to that final line, it would be 5 years gone down the drain, and your PhD thesis committee might be complaining about you taking too much time and not actually proceeding with your work. So what the point I'd like to make here is that it's difficult to define what groups of elements are doing together by a top-down editing approach. On the second hand, I think we require a new sort of conceptual scheme that allows us to make sense of complexity. And what I mean by that is that we'd like to make sense of concepts such as separation of function, right? So are there certain elements that are required for one aspect of specifying regulation, but there's different sets of elements that specify another aspect. We want to be able to make sense of the difference between necessity versus sufficiency, right? So are there elements necessary to specify a particular function, which you can understand by deleting elements, versus asking questions like, what's a group of elements that are sufficient to specify a particular behavior? And lastly, we'd like to make sense of what are called emergent properties, which are widespread in gene regulation and biology at large, which is understanding the interaction between elements. So are there properties of elements that you can't predict from studying individual elements alone, but you can only study by mixing and matching two different elements, right? And thankfully, the way we went about this was, complexity is not a novel problem in biology. And so where else has this been solved? And we thought that it would have been particularly well solved in biochemistry using in vitro reconstitution. So to illustrate this, let's go back to the turn of the previous century when the hottest topic in biochemistry was understanding how muscles contract. Now, one approach that you can take, and which a lot of people did, and I think is extremely important, is one of top-down reductionism, right? So you take your muscle and you fractionate it down into its smallest molecular components. And you can get all the way down to actin and myosin that help muscles contract. But this cataloging doesn't really tell you how muscles contract. And so what you need to do, once you're done with this cataloging and you know what is in a muscle, and I think we're getting to a point now, we know what are in genomes. What another group of people did was take this approach of bottom-up reconstitution, where you purify these components, which allows you and observe them in a dish, and when you or a test tube, and if you purify these components, you can immediately get separation of function because you're isolating them. Secondly, you observe their behavior directly. So whatever's in that tube or slide, that is sufficient to specify the behavior in question. And lastly, once you've studied individual elements or components of your system, you can combine them and try to understand how they work together. Are there properties that they exhibit when put in contact with one another that they don't exhibit when they're on their own, right? And this has been really powerful in biochemistry and especially in the cytoskeleton field. And so we wondered, can we take this similar approach and do regulatory reconstitution? What would we need to do with this, right? What would we need to do about this? So we thought there were two major components of in vitro reconstitution that made it really powerful. One feature was that you have exquisite control over all components in the system. And so to do that for gene regulation, we'd need to be able to manipulate DNA at the relevant scale, which in mammalian genomes is often larger than 100 kilobases at a time. Maybe, you know what I'll do? I'll skip this for the interest of time and say that historically it had been hard to manipulate large pieces of DNA at the same time in mammalian cells. And this is really hard to do with a top-down editing approach, as I've told you. So just as an example, if I had this piece of text here and I wanted to make one change in this piece of text, then editing would definitely be the way to go. But what if I wanted to make 2 changes, 3 changes, 5 changes, or replace the existing piece of text with a completely new piece of text? Then rather than using a top-down editing approach, you might want to use a bottom-up writing approach. And that's the approach that we took to be able to manipulate DNA at the relevant scale. So in our lab, we had, for the last several years, been involved with the synthetic yeast project, which is part of a broader field called synthetic genomics, which seeks to build complete biological genomes from the ground up. And since the early days of Khorana's synthesis of a tRNA gene, the amount of DNA that we've been able to synthesize and introduce into living organisms has seen an exponential increase. And this synthetic yeast project that I told you about seeks to build a 12 megabase genome that has one change every 400 base pairs. Now, I would say that that's a density of changes across a very large DNA space that would be near impossible to do using top-down editing approaches. So we wondered, can we use this technology and apply it to manipulating large sections of mammalian genomes? Secondly, the other feature that we wanted was that we wanted to capture from in vitro reconstitution was one of isolating it from the endogenous context. So you're kind of separating away all the noise and extraneous bits that are going on within the system. So you can focus on what's sufficient. So let's think about how we might do that at HawkSave, right? So just to remind you, this is what all the complicated stuff I told you about HawkSave regulation abstracts down to this, right? So there's 3 things that are responsible for controlling the response of HOXA to these differentiating signals like retinoic acid. Inside the cluster, there's transcription factors like the retinoic acid receptor that bind. Secondly, there's outside of the cluster, there are these distal regulatory elements called enhancers. And lastly, there's some role of genomic context and three-dimensional organization or topology of the DNA that helps to control the response. Now, our question was, what are the relative contributions of each of these regulatory modes? And so to do this, what we decided to do using our ability to build these big pieces of DNA is we built variants of the endogenous HOXA cluster that had different combinations of these regulatory elements, right? So we built one which had both enhancers and these internal binding sites for the retinoic acid receptor. We had one where we removed the enhancers, one where we removed both the enhancers and the internal binding sites, and one where we kept the enhancers but removed the binding sites. So by mixing and matching, we could try to identify the relative contribution of each of these. In reality, these constructs range from 130 kilobases on the bottom there, the smallest one, to 170 kilobases. And then once we have these in hand, what we're going to do is take these clusters from their endogenous locus on chromosome 6 and move them to a completely ectopic locus on the X chromosome. And what we can do now is ask, what are they sufficient to specify, these variant clusters? What are they sufficient to specify at this new genomic location? And by doing this, we think we can get this idea of separation of function because we've built different combinations of these regulatory elements. We're directly testing for sufficiency because we're introducing it to a new location in the genome. And lastly, by mixing and matching these regulatory elements, we might be able to get a handle on what these emergent properties are. So for everything that I'm going to tell you about from this point on, we're actually building rat HOXA clusters and putting them into mouse. And that's because rat and mouse sequence at HOXA are about 10% sequence divergent, which means we can use the SNPs between the rat and the mouse to tell the two apart. And what we can then do is take our rat synthetic clusters and put them into the mouse genome and do all of our assays in cells that contain the perfect internal positive control, which is the endogenous cluster, right? So no matter what we put our cells through, we always know what our ectopic cluster is supposed to behave like, which is the endogenous cluster. So how do we, in practice, actually go about building this? So this is the HOXA cluster. It's about 130 kb of DNA. To save costs of DNA synthesis, which can still be quite expensive at the 130 kb scale, we segment this 130 kb sequence into about 35 kb PCR amplicons that overlap by about 100 to 200 base pairs at their ends. Now, in the case that we wanted to make a change at any one of these locations, we would just replace that wild-type PCR amplicon with a synthetic piece of DNA that encodes for that change, right? So you could introduce as many synthetic pieces of DNA as you want in this construct. We generate these PCR amplicons from BACs, or bacterial artificial chromosomes, that were remnant from early sequencing projects, genome sequencing projects. We get these amplicons, or mix of synthetic DNA, and we chuck them all into Baker's yeast, which is really good at homologous recombination. And it stitches all of these pieces of DNA together to reconstruct the HOXA cluster. We can then recover these constructs and amplify them in bacteria so we can generate large amounts of this material, and then finally deliver it to mouse embryonic stem cells. So we use a fairly complicated strategy to get these big pieces of DNA onto the X chromosome of mouse ES cells. And I'm going to skip over the details, but I'm happy to answer any questions if anyone has any of this. But what you can see is when we sequence our mouse ES cells that arise from this complicated delivery strategy, you can see that they contain the entire sequence that we wanted, right? So in purple, I'm just showing you DNA sequencing traces of purified DNA from bacteria that contains these big 130 kb constructs, and you can see coverage all over that sequence as expected. But now when you introduce them either into wild-type mouse embryonic stem cells or mouse embryonic stem cells where we've knocked out homozygously the endogenous hoxA cluster, you can also see that entire coverage. So it seems like we're able to get these big pieces of DNA in single copy exactly where we want them and how we want them, which is really powerful. Now what we can do once we have these cells in hand is we can put them through this in vitro differentiation protocol that I told you about to see what they behave like, right? So just to remind you, we go from mouse ES cells where no Hox genes are expressed to treating the cells with retinoic acid, which is a developmental signal that cells would normally experience in the embryo to make these motor neuron-like cells. This was in collaboration with Melissa Bulayich and Esteban Mazzoni, who are also here at NYU. And what we're supposed to see in response to retinoic acid is the cluster splits into an active and inactive domain with A1 through A5 expressed and the rest of the genes not expressed, okay? So let's see what happens. So what we initially did was just do a RNA-seq time course at different points of exposure to retinoic acid. And what you can see in this PCA plot is just grouping samples by similarity, is that the samples largely group by time. No matter what synthetic ectopic construct that we put in there, the samples largely group by time. So what this is telling us is that the global sort of transcriptional changes are pretty modest, and we're able to do all of our engineering while maintaining the potency of our stem cells. Now we can zoom into the synthetic Hox cluster and look at how it's responding, right? So this on the left is the 134 kb cluster that does not contain any of the distal enhancers, but contains the internal retinoic acid receptor binding sites. And to our surprise, what we found was that this ectopic cluster, without any distal enhancers, remember again, it's in a completely new location in the genome, was able to specify exactly the correct subset of genes, right? So this is again RNA-seq, where the size of the bubble just tells you how much more expression you're getting relative to time 0. And what you can see is you get A1 through A5 expressed and none of these posterior genes expressed. So it seems like you don't really need these distal enhancers to specify which genes are active. Now, when you look at the larger construct, which contains the distal enhancers, it's also able to specify A1 through A5 with none of the posterior genes expressed. But what I hope you can appreciate is, especially at these early time points, you're getting much more expression. So the size of the bubble is much bigger than the construct here. So what we're concluding from this is that distal enhancers don't specify which genes are active, but boost the amount of transcription that you get once the active gene set has been already set. Now, somewhat puzzling to us, which we haven't fully solved yet, when you zoom out at a later time point in differentiation at 96 hours, both of these constructs look fairly similar, which we're like, what's happening? Is this minimal construct catching up over time? And we're actively trying to solve this problem right now. And if you have any ideas, I'd be happy to hear. Because these constructs were integrated in cells that have the endogenous cluster intact, we can now compare how well we're doing compared to the endogenous Hox genes, right? At any of these differentiation time points. So what I'm showing you here is just RNA-seq data again, except now we're comparing to the endogenous cluster. So if we were doing just as well as the endogenous cluster at every single time point, you would expect all of these bars to be at 1. And what you see is that in both constructs, no matter whether you have enhancers or you don't have enhancers, we basically don't really recapitulate the endogenous expression to the level of the endogenous expression, maybe except in maybe like the 96-hour time point. And what this is telling us is, yes, we can get some expression, but there's still some things that we're missing. And the cool thing about reconstitution is that there's no limit to how much reconstitution that you can do, right? And so one of the active areas that we're progressing now is like, can we perfectly recapitulate the endogenous expression at this ectopic region by building bigger or more complicated constructs? I think maybe in the interest of time, I'm going to skip this slide too, but I'm happy to talk about if anyone's interested, which is just talking about this feature of Hox gene expression called collinearity. And I just want to touch on a little bit about what the chromatin profile looks like at these ectopic clusters. So in undifferentiated cells, both of these clusters recruit repressive K27 trimethyl marks, just as they would at the endogenous locus. In response to retinoic acid, we should see a boundary here between HOXA5 and 6. And what we see when you treat the cells with retinoic acid is, again, a remodeling of the cluster independent of any distal enhancers or endogenous genomic context. You get less H3K27 trimethyl here and more here in the posterior domain, and you only get active chromatin marks in this anterior domain, as expected. And this is also true for the larger construct, the one with the enhancers. Now, if you zoom in, I hope you can appreciate the difference between these two. And it seems like this forms a much cleaner chromatin boundary than this construct here, which maybe points to the fact that transcription, the increased transcription that we see at this cluster helps to resolve the chromatin boundary by maybe clearing the ancestral chromatin state. We don't really have any direct evidence for this, but that's kind of a potential explanation for this result. Lastly, we can zoom in and look at the three-dimensional organization of this cluster using methods like Hi-C. And these black lines are computationally called, it's an unbiased algorithm that calls topological boundaries or domains in these cells. And I've just drawn the blue line to highlight it, but this is not done by hand. And what you can see is this ectopic cluster with the enhancers is able to form a single self-associating domain in ES cells when none of the Hox genes are expressed. And amazingly, when you treat the cells with retinoic acid, it forms two domains, one that contains the enhancers and the active genes, and another domain that contains the inactive genes. So it seems like you don't really need the endogenous genomic context to self-organize in 3D, just as we see at the endogenous location. Now, lastly, what happens if we look at these two constructs where we've kind of completed these four, right? Where we either delete both the distal enhancers and the internal retinoic acid receptor binding sites, or keep the enhancers but delete the internal binding sites. And what we see is that if we remove these four binding sites for the retinoic acid receptor, we completely abolish any response to retinoic acid for these clusters. So on the left is data I've already shown you before, where we have these internal binding sites, and you see the splitting of the cluster into an active and inactive domain. But that doesn't happen anymore. If you just remove four sites, it stays heterochromatinized the entire time. And if you look at gene expression, this on the top is data I've already shown you before. But removal of these four sites is sufficient to completely abolish any expression of anterior Hox genes. And what's amazing is that if you bring back the distal enhancers into this context, it's not sufficient to rescue the gene expression, right? So it really seems like the internal retinoic acid receptor binding sites are the primary sort of mode by which the external retinoic acid signal is translated into a response at HOXA. And so what we think we can do is, using this kind of approach, make these kinds of dose response tables for different allelic variants at any locus of interest, right? And this is just summarizing the data that I've showed you already. And using this, we can build definitive models for how gene regulation is organized at native mammalian genomic loci, and actually start thinking about how these come together and what the relative contributions of each of these are. And so at HOXA, what our studies are indicating is that the internal retinoic acid receptor binding sites are the primary mode by which the retinoic acid signal is translated. These distal enhancers boost the amount of transcription that you get, and once the set of active genes have been set by the retinoic acid receptor binding, and the genomic context is not all that important. So I'm going to skip the conclusions to tell you a little bit about what's next, right? So what can we do with this? So one prediction of our model is that the binding of an activator in SynBYSS, which is at a Hox cluster, is sufficient to induce the response, right? And we've shown this using this data here, where at a wild type locus, you get the splitting into two domains, but if you delete these retinoic acid receptor binding sites, you don't. So we thought, what if we go into these sites and replace it with binding sites for a completely synthetic transcription factor that can be induced by a completely orthogonal signal, right? So for those of you who've used RTTA, the TET transactivator in the lab, this binds to a 19 base pair motif and is induced by a signal like DOX, a small molecule, that's orthogonal to the rest of the genome. It doesn't really do anything to the rest of the genome. And so what we decided to do was build this TET-O SynHox cluster, where each of these binding sites is replaced by a TET binding site. And now what we can do is say, what if I leave out retinoic acid from the differentiation, but just treat the cells with DOX? Am I going to get expression of the appropriate genes? And this is what we see, right? So now looking at, so we did 3 different treatment conditions. One, retinoic acid, just as we did normally. One where we have both DOX and retinoic acid, and one where we only have DOX, right? So now we're looking at the endogenous HOX genes, and you can see that they come on in response to retinoic acid, but don't really come on in response to doxycycline. This is what we expect, right? But this is the amazing thing at the TET-O HOX. The TET-O HOX does not respond to retinoic acid, but now it does not respond to retinoic acid, but now responds to doxycycline, as you can see here. And the original ectopic cluster that I showed you responds to retinoic acid, but doesn't really respond to doxycycline. So we think this is quite an amazing result, where we've been able to kind of rewire the inducing signal of an endogenous genomic cluster by using a completely synthetic transcription factor. And what we think this is an orthogonal platform to study how this works, right? So because we're using a completely synthetic transcription factor that doesn't have the effect on the rest of the genome, we can do all kinds of things that you wouldn't be able to do with native players, right? So we have complete control over the transcription factor, the binding site, as well as a small molecule inducer. And so we can change transcription factor domains as we want to. We can change the amount of signal that we add to the cells. We can also change the binding affinity of the DNA binding site of these transcription factors. And so this is taking a fully synthetic biology approach to rewire endogenous circulation to understand how it works. This is kind of more for the genomics people at heart. What we can start to do is also manipulate the relative position of endogenous transcription factors, which are thought to set this chromatin boundary. And so we've built a lot of these variants now where we've manipulated the relative position of the retinoic acid receptor and CTCF, which are thought to be the 2 main players in setting which genes are active and which genes are inactive, which again, I'm happy to talk about in the question and answer if anyone's interested. And lastly, what we can do is we're not just limited to studying the HOX-A cluster, right? We can study any locus in the genome. And so we have active collaborations going to study the Ixin Activation Center. This is a pretty crazy project to understand the basis of mole intersexuality, and also maybe creating fully synthetic regulatory domains from the ground up in organisms like Drosophila. Most importantly, this is now in the context of a large project called the Dark Matter Project, and you can visit our website, just the darkmatterproject.org, to learn more about how we do this. And there's a link that you can click to submit projects that you want to maybe collaborate with us on and work together. With that, I'm going to thank a bunch of people, specifically Jeff Buca, who is my PhD advisor, and Liam Holt, who is a close co-advisor, as well as Esteban Mazzoni and Milicia, who are our collaborators to understand who really were the HOX brains behind this project. I'd also like to thank our funding sources from the NIH, you for your attention, and Taesuk for the invitation. I'm happy to take questions now. Thank you.
Oh, fantastic, fantastic talk. I love it. I wanted to ask whether Mike has any question.
Yeah, that's very cool. I was just thinking about it in the context of the human genetics projects that I've started working on. And as you know, human genetics gives us statistical associations, and we have hypotheses about what are the causative agents, but really genome editing is the way that we can prove which factors are actually, which variants are actually causative. So
Yeah.
sometimes that might be possible with a point mutation, but I think more generally, the field of human genetics is going to depend more and more on being able to do genetic manipulations, including these large ones. So I think there may be a lot of possibilities there.
Yeah, no, absolutely. So I think that's a great point. And so the way we initially thought about this work was there's two kinds of angles that we can take with this, right? So one we're calling synthetic hypervariation, which is kind of the work that I showed you now, right? Where you can take an existing locus and vary it in all kinds of ways to understand how the regulation works. So manipulate enhancers, transcription factor binding sites, things like that. Now, the other thing that you can take, which I think you are absolutely correct, is to build synthetic haplotypes, right? So if we have from GWAS a whole bunch of risk haplotypes, we don't often know what the causal SNP is. And if there's multiple SNPs working together that need to give you that change in expression, that's then ultimately causing the phenotype, not to mention if you're looking at complex disease, right? So what we're in a position now to do is start building risk protective and maybe variant haplotypes to try to understand what are the causal SNPs or series of SNPs that you need to get change in gene expression. So there's quite a few projects underway in the lab, none at the level of publication yet, unfortunately, but I think we'll learn a lot from that as well.
Cool.
Any other question or comment, Mike? Okay, so let me ask one question. So that's super cool work. And, I thinking about the history of whatever molecular biology or synthetic biology, to me, more than almost 50 years ago or more, actually 70 years ago, we've, discovered DNA helix structure, and then I would say that is basically our start of learn alphabet, and then the DNA sequencing, by multiple people, including Mike, Brent. That's basically we start to read whatever sentences, and then DNA synthesis basically giving us ability to write, but not necessarily really creation, but that is basically copying whatever already written by nature, but we have the ability to write. And then CRISPR or genome editing tool, now we can correct or edit, but not necessarily we really understand the meaning of the writing done by nature, or we understand some, but not a whole lot. But your work, I kind of now see we start to understand the meaning between lines. So that's kind of exciting to me. But still, to truly write something, there are huge challenges because we still not that much understand the real meaning between whatever text. So could you kind of give us the remaining challenges and opportunity for the next 20 years? And that I want to hear from you.
Sure. Yeah, so you're absolutely right. We're not doing creative writing with DNA, right? We're shamelessly plagiarizing from nature, as I like to say. And so I think the fundamental obstacle is not one of technically being able to build big bits of DNA. I think the fundamental obstacle is knowing a set of instructions that make sense to a cell to behave in a particular way, right? And I think the way that I think about going at the first step of that is, so let's assume you didn't know English, right? And but you had access to a machine that if you gave it a series of English words or sentences or paragraphs or books, it would tell you whether it made sense or not, right? It would tell you whether this computed, let's say. Now, one way I think that's pretty intuitive for people to understand then how English works is just to feed random bits of text to that machine and figure out, okay, these sets of things made sense and these sets of things don't make sense. And if you do it enough times and you iterate and you learn, slowly you can sort of decipher the rules by which English works, right? You might not necessarily know the words specifically, but you know, we can, that's a complexity that we can get to, but you'll know the grammar of how English works, right? So we have access to such a machine when it comes to biology, right? We have cells that can take a random message and tell us whether that message makes sense or not.
Right.
So I think what we're in the business of is just typing gibberish for the next 5 years and seeing what makes sense and what doesn't. And if we've blanked out enough of that space, well, whatever is left, we'll be able to then use that to kind of map out the grammar by which gene regulation works, right? Or at least that's how I think about it. Yeah.
Oh, I love it. I love that answer. Yeah, that's true. Actually, we just constantly providing whatever random sentences, and the cell will figure out. I mean,
the cell knows, right, whether it makes sense or not. And
right.
we have
we still sort of have some grammar. We understand, you know, the promoter should be in front of gene rather than after gene. We know the basic grammar, and then we just kind of providing that whatever sentences, you know, inspired by the native situation or system, and then we But
I'd say I would argue that we don't often know what a promoter is, right? I think in bacteria maybe it's a little bit more worked out, but in mammalian cells, for example, like I don't know what a promoter is. I don't know where it starts, where it ends. I don't know what these regulatory elements are. I don't know how many of them I need. I don't know what order I need them. Do I need to have a particular spacing between them? I don't know any of this, right?
Exactly.
And I think what the approach that we're taking is saying, let's just do as much of it as possible. And now we have tools to, you know, very careful detail, understand whether a particular piece of DNA makes sense to a cell or not, and just do it over and over again and ask the cell, hey, does this make sense? And most of the time it's going to say, no, it doesn't make sense, or it makes sense in a way that you weren't expecting. But if you do it enough and you do it systematically, I think we'll be able to maybe get a handle on this stuff.
I love it.
See you, Michael.
Okay, thank you so much. That's a wonderful answer. You know, I actually start my lecture kind of giving bunch of random ATs, GC letters, and then to make the student figure out. Also, I kind of make random English sentence. I kind of ask them to figure out without any space between the sentence. I mean the word. And then student figure out, because student know the grammar, but cell somehow know how to deal with that random sequence. So I love your answer. That is probably the best answer to me so far. So thank you. I love it.
Thank you. Appreciate it.
Because I'm asking that question. So the question I'm asked, that question is asking to me every single day. I mean, literally. So okay. So let me, I have the question from audience. Okay, so Ryan asked question. That's comment. Basically, thank you. I have to learn, but this was all very interesting. Okay. Right. Answer right on. Okay. So that's wonderful. So I will cross. Let me see. Yeah, I'll cross. Okay. So thank all for joining and staying today. We'll meet again next week on December 15th, Thursday, the same time, the same Zoom link. We'll have Dr. Scott Wolfer, the Science Director for Synthetic Biology at Office of Naval Research Global, and Professor Anab Mukherjee at UC Santa Barbara. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. And thanks. I will stop recording. Just give me 1 second. Stop recording.